Okay, so Marty's going to switch laptops. I will introduce Marty. I've already humiliated him some, I think. So his job is to humiliate, who comes next? Doug, is that right? So Marty, many of you may know Marty's papers from the 80s and 90s, mapping visual areas. He has had a long history as a neuroscientist. But what you may not know about Marty is he also loves to hack. He wrote a lot of the code that became Freesurfer, a lot of Tiki Surfer in particular. He evidently still has the original code base and maintains his own version.
Freesurfer Classic.
Freesurfer Classic, yes, Freesurfer Classic.
Okay, so this is a scientific talk, but I'm going to go so fast with the slides that you should think of it just more like a little tour, a little video tour. So it's kind of a personal history of Freesurfer and how Anders and I initially sort of came to the ideas of trying to do this. And it started off with turtles. So I always show the same slides, but I don't put any labeling on them so that I can give different talks. So it's turtles all the way down. So what about turtles? Well, I was always interested in topological maps. And I started off studying the attention system of a turtle. You probably don't even think a turtle has attention, but they actually do have attention. This is, if a turtle drinks coffee, this is the system that's turned on. And that's a beautiful terminal from the superior colliculus of the turtle that shows this map in the superficial layers. You can see it's like 1/10 of a millimeter across, so it's a very fine-grained map. And there were some non-topological maps in turtles, but topological maps are the first principal component of organization of the brain. And you might not have even realized some of the places that there are maps. So this is a picture of 2 injections into the caudate nucleus right next to each other. And you can see when they project down to the substantia nigra, they're topographic. The ones going to the GABAergic part of the substantia nigra are topographic over there, but the ones going to the dopaminergic part are topographic. And if your index finger does something good, why reward the middle finger? Why shouldn't there be maps, maps of reward? I'll show you little pictures of the cerebellum later. Here's some of the structures that project to it. If you look at it, the inferior olive, you can see it's a jammed up little cortex in there. That's a cross-section of it. Here's the output. It's another little cortex in the dentate nucleus that's maintaining a 2-dimensional map. So there's just maps all over the place. And so I'll just take you over a quick tour of some of the things that I was working on right before Anders and I started writing the first code for what became Freesurfer. So, why does the visual system have all this stuff that we study, like blobs? It's for eating bugs at night. That's what it all came from, sad to say. So I studied the owl monkey. It doesn't eat bugs. It's more of a fruit eater, which I eat a lot of fruit now. And I spent many days. So like Monday is where the bee is, and then Thursday is down sort of. I arrived at auditory cortex on Thursday, marching across the giant deserts of visual cortex. If you look at a section of that brain, you can see that there are a number of different areas that you can see in a myelin stain. But we always had the feeling like if we could just flatten it out, we would be able to actually see some of these things in more detail. And so there's a small but select group of the Flat Brain Society who really wanted to flatten things. And here's a beautiful flattening of an owl monkey. And so you can see some of the major areas like MT and the V1 border very clearly. V2 is a little harder to see. If I sort of give you a little after image, you can sort of see it. But the way that you really have to find most visual areas is by mapping. So there's a bunch of visual areas right there. You've got to map in extremely high density. So that's a millimeter down there in order to actually pick up these areas. You also have to stretch things because after we flatten the cortex and stuff isn't lined up. And so this is some 2-dimensional morphing software from the really old days in order to actually take a penetration photo and warp it into what it became after I flattened it. And so I developed these methods of trying to distinguish different visual areas by their visual field sign to analyze this invasive data. These areas are quite small, like I mentioned. So here's one that's actually narrower in a tangential direction than it is in a vertical direction. And if you look at something like a mouse, they have these tiny visual areas. They represent the whole visual field. They're 0.05 square millimeters in area. So it's just a tiny little tube that's got a map in it this way that extends through the cortex. Some of these things are correlated with, there's MT correlated with myelination, but unfortunately that's still MT. So you really have to do mapping in order to pick it up. For some reason, the periphery of MT gets less myelinated. They vary a lot between individuals. So here's 2 different individuals at the same scale. You can see that area varies by almost a factor of 4. So that really requires some kind of method of doing it with individual subjects. But you might wonder, what about all those little blobs and patches? Whenever you inject in the cortex, you see these little patchy things that people like to study, like, and Roger has some fine recent papers on V2. They're everywhere. So if you go to S1 or M1 or V1 or prefrontal cortex, they're there. But 90% of the connections are within those little circles. And so that's the second principal component of the blob. These little patchy things is the second principal component. It turns out that our standard voxels are just about the right size to pick these things up. And so when I first got to UCSD, I was trying to do monkeys. But UCSD more or less made that impossible. So I would drive up to Caltech. This is a fat-tailed lemur. You can see that's actually quite a bit smaller than an owl monkey. But that really sort of emphasized, I always had a comparative approach, and this really sort of emphasized, looking at a lot of different animals, of which, humans were one, including a squirrel. This is actually an actual photo. Somebody was taking a picture, and they set up the camera, turned on the timer, and the squirrel popped up in front. There's a squirrel cortex compared to a rat. So I did some work on squirrels, and for Jean, I thought I would just show her the nice verrucae in the entorhinal cortex of the squirrel. You check out the rat, nothing in a rat. Squirrel's really high-quality animal. Nice visual system. But I was really interested in humans, because I originally did my PhD in the philosophy of science, and I was really interested in sort of, dealing with that. The problem at the time is we didn't have any way of easily doing that. So I took a direct approach. I'm good with my hands. And so, so I just spent, this is about 12 hours, carefully peeled off the cortex. And so I knew it could be flattened because I had actually flattened it. It wasn't super flat, but that's the calcarine sulcus. So that's why we would cut it this way, because this was the very first cortex that was cut that way in the 80s. And I found something that looked like MT. I called it MT, but it turns out MT is down there. But Roger made the same mistake. So at the time, structural MRI didn't work that well, especially 3D sequences. So this is the modern scanner that I had in London, but at the time, in 1990, 3D sequences didn't work very well. And although Anders and I sometimes got lost hiking, Anders isn't here. He got quite stuck up on one very cactus-laden hike, and when we got lost, but he never got lost coding. And as Bruce mentioned, the origin of this cortical surface software was for EEG-MEG constraints to basically deal with this kind of problem, where if you have the cortical surface, you can, rather than doing it in a volume, you can actually do a better job of trying to localize things. And it's got nasty things that make it difficult to directly use the surface normal constraint, and people are still working on this very intently to try to take full advantage of this. But at the time, I just wanted to sort of show this historical slide. The 3D scans didn't work well enough, and so we actually had to construct a high-resolution scan from thick slices in different planes. And since Anders was already doing all these, Anders and I were doing these linear inverse things, it's just another linear inverse where you've got these needle voxels and you essentially make a forward solution and invert it. So then there was a burst of coding. This was Anders' standard form, like, remove all comments, say something once, why say it again? So this is the first surface made from that very fine quality inversion and recovery scan. So the contrast was fantastic, much better than an MPRH, and that actually helped make this possible. So you can see these are pictures taken off the screen like we used to do in the old days. We had to actually sort of set up a camera in front of the silicon graphics. And we made this video in 1992, the original pumping brain video. And I had this ancient video which I couldn't actually recode because it was a beta cam that we showed in 1992. But this was the one that Bruce shows. This is what explode flag does. And I remember showing this in Anaheim, and Eric Schwartz came, and he thought. Well, we were young, we were young, and he was a little older at the time, and he thought that it was a little bit unseemly for the field of brain flattening. So that's the original explode flag. It's just, it's under standard form. It was only like 4 lines.
Which are kind of invented, right? The field of brain flag.
Yeah, absolutely. Yeah. So I mean that's why we were a little sort of brash young people at the time. So by 1996, we had hand reconstructed. This is in the days before topology correction. Hand reconstructed a number of brains. These came from Germany. There were some from Münster and a lot of other places. And we immediately realized there was a problem because they all look kind of different. And so I thought I would turn it into an ellipsoid, not being as good a programmer as Anders. This was my first attempt at an ellipsoid. But I got the ellipsoid working eventually. And it was kind of a pill-shaped ellipsoid, which distorts the cortex less. There's a lot of nasty properties of dealing with that. But this was the first morphing video of actually sort of stretching one brain into another brain on an ellipsoid. And it has the big advantage that you can actually line up a sulcus with a gyrus, which we considered to be a really big advantage at the time. The field did not. The field was kind of upset. I remember we got a lot of heat for this in the beginning, but then you could apply a coordinate system, even on the ellipsoid. Yeah, and that's nasty code. Then Bruce came along and rewrote everything. And the code got a lot bigger. The under standard form code is it's very short. I think the original Freesurfer code is probably somewhere on the order of 20, 30K lines. It's bigger now. It's bigger and beefier and much more robust. But one of the big advantages is that you can deal with all these little crinkles. So you can see them right there in the flattened cortex. You can see there's all these secondary crinkles. And one of the things that, especially in interacting with 3D based analysis methods, if you surface average the surface coordinates, you get rid of all those crinkles and you get a shrunken head. So it has quite a bit less surface area than the. So if you average the inflated ones, then you get a normal size head. It makes sense to use this if you're doing 3D stuff, but we all know that's for the cortex. We want to use the surface. So what about actual mapping? So the whole time I really wanted to find all these visual maps in humans. At the time, MGH had by far the best scanners. And I remember years ago, I went to a talk that Brian Wandel was giving, because Anders and I had gone up to San Francisco to play around with the spiral scans they had running there. And then we went out to MGH and we said, oh man, you got so many more slices. You had 8 slices at the time. They only had 2. And then we talked to Ken Kwong at about like 3 in the morning. He said, oh yeah, just take off this one parameter and you get 16 slices. And you got like, whoa, for nothing? You could just suddenly get 16? I walked into a talk years ago that Brian Wandel was giving, and he sort of wheeled around and said, you should have come out and worked with us. And we said, well, they had more slices. And so over the years, I did a lot of work on extending things to higher areas. This is some work that Anders and I, a failed attempt to try to map dorsolateral prefrontal cortex with Tommy Vaughn's shielded coil looking through the little mesh. Yeah, that was late at night, didn't work. But we just added a little sort of distractors, which are actually stimuli, and then it worked, and it gave us an LIP map. And one of the things from having a comparative background, I wasn't at all upset by the fact that LIP, which is supposed to be lateral interparietal cortex, was actually medial, because it just sort of gets pushed around. I'm used to sort of morphing things and seeing things morph across species. The field didn't like that, but eventually they sort of got around to it. Actually, a lot of people thought of parietal cortex as this kind of default mode network as opposed to the LAP that people knew from macaque monkeys. And then we went to sort of using windowed videos and other things that were more interesting, or this particular nightmarish thing where you monitor for upside down faces and an occasional number to keep your attention up. And so what you're mapping is not really retinotopy, you're mapping attentionotopy. There's only one true color scale for retinotopy. The red is upper, green is lower. So over the years, we did quite a few maps of different people, but we eventually ended up doing complex valued retinotopic averages on the surface using Bruce's fine MRIS register. And if you do that, you can see that that's our original LIP. But you can see quite a good chunk of the cortex is retinotopic, including a bunch of stuff up in frontal cortex. And there's lots of LIPs, lots of VIPs and LIPs. We did a lot of work on trying to find some of these multisensory areas. So if you look at the boundary between somatosensory and visual cortex, you find these multisensory areas there. So we made this fine device to gently puff the faces of the CogSci graduate students. And you can actually see a face map in this multisensory area in parietal cortex that really has a retinotopic and a somatotopic map that are superimposed. And it turns out to not actually be a retinotopic map, it's a face-centered map. So if you move something around the face but look straight at it, so you're not changing the retinotopic stimulus, you actually see that it's a head-centered or a face-centered map that's remapping visual things into face coordinates. So it's really, you can think of it really as a somatosensory area in a way. There's actually 4 of them probably if you average across people. And then we went on to develop the air puff bodysuit. We avoided some of the sensitive bits, but 64 channels. And it turns out there's a map all the way down to the toes. So it's a weird little homunculus. It's got you can see it's got a face and an axial body all the way down to the toes, overlaying a visual map, and then the movable hand is off to the side in lateral interparietal, or in anterior interparietal cortex, lateral to lateral interparietal. But again, from a comparative point of view, you get hind limb movement when you stimulate that place in a bush baby. So this is it's a very preserved kind of part of the cortex. In the early days, we did tonotopy. Now, auditory maps are different. They have these point-to-plane projections instead of point-to-line projections. But you can do the same kind of tonotopy. And this was with Tom Talavage and Randy Benson originally on mapping the cortex. This was 1996 data. Sadly, we got scooped by 1 year. in 2003, but we were there first. We've gone on to do auditory attentionotopy. So this is regular tonotopy, but what you can do is, if you look in the middle there, you can actually have a spectrally constant stimulus, but have people attend to different parts of the spectrum, and you see the same maps that you see with actual tonotopy. So it's very similar to the visual system that way. And the brain really plays around with maps, especially in the auditory system. So if you look, tonotopy is going that way, but there's another coordinate, characteristic delay, going the other way. And if you actually look, this is in one of the, something like the medial superior olive equivalent. If you look at that map, if you go up higher, you can see a space map is constructed by rotating the point-to-plane projections to generate an auditory map of space. So there's a lot of sort of messing around with maps, and we haven't sort of finished them all. There's probably these two-dimensional bi-frequency maps and a lot of other, there's something perpendicular to tonotopy which we haven't completely pinned down. Okay, so I'd like to now sort of go on to some work in trying to look at quantitative architectonics, again, harking back to these transferring stuff that we knew from animals, but actually trying to do it in awake, alive people. There's various ways of doing this. I was talking to Doug earlier about it. This way that we used here was to take that equation, which is kind of messy, and linearize it. It's I don't know much math, and so that's just amazing that that is the same as that. But if you then do two flip angles and then also do some mapping of flip angle to, because there's a large variation in flip angle even at 3T, then you can actually try to relate this to retinotopy. And again, we got scooped by 1 year by Van Essen, sadly, but our maps are a lot cleaner. So one of the things we found was that lateral spot's not MT, and Van Essen has recently rediscovered this in recent years, but only about the front, the back third of it or so is actually MT. You can see from the retinotopy right there and the ipsilateral responses. Here's another thing that we collected early in the days that Arthur will remember. It's this fine stimulus motivated by the late Ralph Siegel. And so we did this stimulus, and we found this giant spot on the medial wall in probably 1995, 1996. And this is a great example of theory-driven observation. We said, what's that? You know, I don't really know what that is. And sort of put it aside. And then eventually, with some Italians, we got back to actually trying to figure out what it was. And there's a nice hemifield map there. It's very hard to map the eccentricity. So this is a scan where I sit in the scanner for about an hour and a half with my head tilted to the side with the screen about 2 inches from my eyes in order to be able to map the periphery. And if you do that, you can see it's got a beautiful eccentricity map that starts at the place where people leave off in their retinotopic mapping. And it actually corresponds to that heavy myelination. So it's an area that we also know from primates that's very sensitive to these flow field type stimuli. And then we found V3A. That was the one that both Roger and I found originally and thought might be MT, but it wasn't MT. So one of the advantages of quantitative approaches, which people are working on heavily here as well, is that if you start with better data, it's easier than fixing it later. So you can sometimes reduce the amount of code that you use if you start with better data. I've developed a lot of respect for the MR physicists over the years. It's always better to start with better data than to try to fix it afterwards. Another thing that has really been motivated by the original comparative approach to architectonics was to try to use diffusion to try to pick up these textural differences between different cortical areas, because that's how the original architectonicists distinguished cortical areas. And some of them got a little crazy. Here's a comparison between the columnar organization of the brain and the Parthenon. You get a little crazy when you're an anatomist. But it turns out there's a little bit of gray matter diffusion, and it's repeatable, and you can actually use it. If you take Hardy data, you can actually try to generate features relative to the local surface normal to try to look for difference in diffusion patterns between different cortical areas. And there's some rotationally invariant ones too. This is way overdetermined, but we just want to make sure that we covered all our bases. And you can use it to recognize or train and recognize areas on a test-retest, or you can do unsupervised learning on this. So just try to k-means cluster, completely unconstrained k-means cluster some of the diffusion data. And so if you look at the T1 data and compare that to k-means clustered diffusion data, you can see there's more information in there. And there's even more information if you use several b-values, sort of naughty type things. So you probably got an extra, you're recording 60 directions, 3 b-values. That gives you another 2 or 3 dimensions, something like that, past your T1. But I was a philosopher originally, and so do these maps have, are they good for anything? So we did frontal mapping, this fine experiment where you try to do a face 2 back while fixating the center, extremely irritating. But if you do that, you can see frontal maps, those same kind of frontal maps that I showed you in the retinotopic average. So dorsolateral prefrontal cortex is mapped, even the one that doesn't care about location, it's still mapped. And if you have somebody imagine moving around in a building versus stopping, you see that activation mostly overlies maps. So these maps are involved in something like imagining, navigating around. But what about language? And so this, Arthur will remember also, the famous soccer goals experiment. So we did a very carefully controlled experiment where we looked at videos of famous soccer goals. Arthur was, I think this is Arthur here. And then compared them to sentences. Give it all, good sentences. They were good sentences. Compared to nothing, compared to fixation. And you can see that soccer goals wins. So the linguists and psycholinguists weren't too happy about that. So and it didn't include other maps. So what we've done more recently is to do all the maps. So do auditory and then even body mapping. So here's an example of what it looks like. So there's some polar angle maps, and here's some body maps where you can see VIP and the supplementary motor areas and S2. And then here's a map of the mouth. You can have somebody sort of articulate different consonants and see a map of the mouth. There's some auditory maps, and then we can just paste all the maps together. So there they are, all together. So it's about half the cortex, a little over half of the cortex, but just straightforward sensory maps on it. And then we can have somebody, read about Brodmann, actually move their eyes like they would in real reading, which is rarely done in the magnet, even though it's quite easy to do. And compared to sort of duration and size matched Hindi transliterations of English words, which are not readable. So you get something like that, and so that's versus nothing, that's versus Hindi, and now you can compare it to the maps, and you can see most of it is in the maps. And so maps are not bad. A lot of times cognitive neuroscientists want to sort of say, oh, it's got a map in it, so it must not be doing something good. So that's not the case. So we've done this better with real multiband data. So I'll show you, this is 20 subjects where we did the same thing to all of them. So retinotopic maps, and you can start to see the dorsomedial frontal eye fields with better signal to noise. And there's the auditory maps. You can see the dorsomedial frontal ear fields, which are attached to supplementary motor cortex. You notice that the frontal visual maps versus the frontal auditory maps are actually mostly non-overlapping. There's our body map's got some motion artifact in it and auditory self-stimulation, but if we take that out, that's our having people actually make movements, very small movements. And then if you do that, you can see that the dorsomedial frontal eye fields and frontal ear fields are right next to supplementary motor. And then you can have them do this again. And then if you look at the language versus Hindi comparison, you see the usual suspects, some parietal cortex and dorsolateral prefrontal and STS and infratemporal. But if you actually look at the overlap, there's, again, quite a bit of overlap. About 85% of the activity induced by actually understanding language in a naturalistic context overlays maps. What about picture stories? So we got these nice children's picture stories and directed people's saccades around. And again, you can see a lot of activity when you compare it to scrambled up pictures or just out of order pictures. That's out of order pictures. But again, if you look at the comparison between all 5 of those different things, you see there's quite a bit of overlap. So there's a few areas that escape. Like there's one little area right there in the STS that escaped our maps. But there's really quite a bit of overlap. And so it's worth continuing to map in relation to cognitive studies. Okay, so for the last little bit, I'm going to talk about some stuff done with my hacked version of Anders Freesurfer Classic. So I wanted to warm up for the cerebellum, and so here's a dolphin. So a dolphin, this was a famous Navy dolphin named Bucky who died. So dolphins are trained by the Navy to attack enemy divers. That's actually what their job is, because they're very aggressive, the male dolphins especially. So it's got a very thin cortex, very strange cortex. So look at this thing. It's incredibly folded up. But if you look at it from the top, it's really squashed. So it sticks out sideways like a tube. And it's got some weird sulci that you never heard of, like the limbic cleft. What's that? And then when you look at it, look where the visual cortex is right here. Auditory cortex is here. This is the insula. Shouldn't auditory cortex sort of be somewhere down here? What's it doing up there? So it's really quite amazing. It's actually bigger than a human cortex because it's so thin. So it's actually got quite an extent. It's a very primitive-looking cortex, but there's a lot of it. I haven't got to one of these yet. The one at the top is a killer whale. That's the biggest brain. It's actually got less neurons than humans, but it's got about probably 4 to 5 times the surface area of the cortex of a human. But this got me in the mood to go after the cerebellum. So 75% of the neurons in the brain are in the cerebellum. So if you look at the cerebellum, you're not easily able to pick up the cortical surface. That's the cortical surface of the cortical surface. Here you're only picking up the major lobules, not the actual cortical surface. So you need a dead one for this. So that's me alive. That's what it actually looks like. So it's just an insane structure. So here's a T2 star scan and a proton density. Here, combine them, add a little special anisotropic filtering sauce. And you got this insane structure, 5 million vertex structure, very difficult to. It actually loads. Freeview will load this. It takes about a minute, but it'll load it. You actually need that kind of resolution. So it's 190 micron cubed resolution to make this out. So that's the size of an actual Purkinje cell along one of these little folia. You can even see there's the dentate nucleus. That's the output structure, a little cortical output structure of the cerebellum. So I tried it the other way. It didn't look as good with the cerebellum going down. So here it is unfolded. It doesn't unfold very well because the way it grows, you get all these little bubbles. So you can see all the individual folia. Those are the little stripes. But then you can see those really deep divisions between the individual lobules. And yeah, it's an amazing, amazing structure. So what to do with it? Well, we needed to sort of chainsaw it because it has to be flattened. I know people don't like flattening things as much, but so we took out the Freesurfer chainsaw, and after about 20 rounds of editing to remove wormholes, sawed it up. And so here's what it actually looks like. So that's the anterior lobule, and then you pass the primary fissure, and then you go back into lobule 5, lobule 6, and then you get into the cognitive cerebellum. This is Crus 1 and Crus 2, and in lobule 7, and then lobule 8, and then it sort of goes down, and then you finally arrive at the tonsils, the paraflocculus. So what does it actually look like? It's this ridiculous thing. It's about 5 feet long. It's about 5 centimeters by 5 feet. So it has almost almost the same cortical surface area as the cortex does. So what is in it? Well, of course, there's maps in it, and so there are these weird little fractured somatotopic maps in there that's like 50, 60 penetrations in 1 square millimeter. There's like a little patch of rat hand and a little patch of furry buccal pad and a little patch of the back. So there's all these little patches. It's so strange that people have hardly studied this. It was discovered in 1978 originally, but people have just ignored it because of just the insanity of the structure. So what is the size of these things? They're like that. So some work remains to be done on trying to understand this. But it's important because when you actually look at the cerebellum, it's expanded slightly more than the neocortex has expanded relative to monkeys. So it's just as much of our humanness as the neocortex is, and it's very poorly understood. Okay, so in the last 2 minutes, I'm going to go over some maps in places you wouldn't have thought there might be maps. And so this was an experiment that I thought for sure would not work, but I agreed to do it anyway, just in case. And so it's a very simple experiment. Just touch people with sandpaper or have them view somebody being touched, sort of a mirror neuron, like mirror neurons gone wild. Except the trick here is to record in early sensory areas. And you can view in first or third person, or you can compare, like, what if you just get close with the sandpaper but don't touch? And also all the people experienced actual touching with the sandpaper in addition to just watching people. And this is some 7T data from Leipzig. So we'll just compare sort of actual touch to viewing touch. And we have to sort of up our pipeline a little bit to do searchlight circular correlation of these complex valued data sets. And we did it fairly. So here's some voxels intersected with the surface. And so we find a voxel that is the nearest one to the average of the ones that got sampled onto the surface, so we don't sort of double dip. And then run a searchlight around and run correlations. And it's S1. So there's actually a visually induced map, either by first or third person view of somebody being touched in S1. Now, it's not a big signal. It's in area 3B and 1. It's not a big signal. So if you look at it, it's like about 10% of the actual touch signal, as it should be, because if you watch somebody being touched, you don't want to like, whoa. You might want to sort of like feel a little bit like I know what I can feel your pain, but you don't actually want to mistake it for a real touch. And it could even be an inhibitory signal, we don't know. But this is even me, Mr. Map, did not expect this one to work. It's in its 10th review now, and it will be coming out very soon. So we did a lot of other things to sort of make the reviewers happy. But in conclusion, this medieval theories of how the brain works, you have the common sense at the front where all the modalities come together, and then you have like warp drive in these ventricles. You know, you have imagery, cognition back here, and memory in the ventricles. That's not right. There's a lot of maps, so don't, this one, especially this one, having a map does not equal unimportant for cognition. And it certainly could be the case that in some of these higher areas, we just don't know exactly how to map them. We know connectionally, they look just like the earlier areas. They have all these extremely local connections. So I'm now back in San Diego State, and I'll just leave you with this fine picture from the early Freesurfer days of turning the color scale to a position that it shouldn't be put in order to just show you places that were modulated by visual mapping. So thanks.